So my lab is in Berkeley, and I'm interested in herpesviruses. And essentially, we're using herpesviruses to try to interact, to understand how they interact with the host immune response. And we're working essentially on herpesviruses because they're extremely well adapted to their host populations, to the humans. They're very ancient, they're host specific, they're so adapted, they're host specific. As you all know, they're all persistent, zero prevalence, and they lead to modest pathology, and that's for people that don't know much about herpesviruses. And what we like is they are large viruses. And that means that they have space in their genome. They can encode for protein that really modulate host responses. So what we're trying to understand is what host responses interact, how they interact, how the cells interact with those viruses, and how these viruses interact with the cells as well. So we're working with different herpesviruses in the lab, but essentially today what I would like to talk, it's a story about Kaposi sarcoma-associated herpesvirus. So it's a story that we studied about 2.5 years or 3 years in the lab, and it's been done by a graduate student, which is now in the audience, which is like Elena Beckerman, which is now a postdoc in Stanford. So that will be her story, and if you have some tough question, that will be for her. So. Okay, so essentially, you heard about Kaposi sarcoma from the great talk from Zoe Davis this morning. That essentially is one of the leading problem associated with HIV. And this virus is essentially associated with Kaposi sarcoma, so these skin lesions on patients. And people that have that, if it's not treated, can die really fast from these infections. It's also associated with B-cell lymphomas and B-cell lymphoproliferations. And the mode of transmission of the virus is not really completely understood, but it's believed to go through the saliva. And like exactly in the talk of Ann Arvin, is thinking that it's believed that those viruses that are in the saliva will infect first the epithelial cells near the tonsillar tissues. And those cells that are infected, these epithelial cells, will then allow the virus to be released and to lead to B-cell infections and later on endothelial cell infections. And the virus will reside into those cells for a really long period of time. Okay? So how does the virus enter the cell? So the virus will get, as you know, released, fused with the cells and interact with the receptors, fuse with cells. The capsid is delivered to the cytosols, migrate to the nuclear pore, and the DNA is injected into the nucleus, where it will circularize. And at that point, the virus has to decide whether it wants to enter the latent stage or the lytic stage. And the latent stage, meaning the viral episome, will be maintained tightly associated with the chromosome, the genomic DNA. There will be really little viral expression at that point, and the only thing that will be expressed in all the cases is one protein called LANA, which is late associated nuclear antigens. That is essentially a protein that has two arms, one domain that will bind the viral episomes, and one domain that will bind histones on a chromosomal DNA, on the chromosomes. And that will maintain the virus associated to the chromosomes. When the cell replicates, the cellular machinery replicates the episome as well, and the virus is then transmitted to the two daughter cells, and this way it can proliferate within the host. In addition to LANA, there is also 17 microRNAs that have been expressed during this latency, and those are believed to modulate the host to allow for maintenance of this latent stage, maintaining the episomes, and also avoiding innate immune responses. In vivo, we don't really understand yet exactly how that, what are the signals, but there is cellular signals, stress probably, or apoptotic events, that can lead to the expression of the RTA protein from a latent stage. This RTA protein is called the lytic switch proteins, which essentially that's a protein, viral protein, that will initiate the lytic cycles. And during these lytic cycles, you will have a cascade of gene expressions. You have DNA replications that we heard earlier, and that will lead to the accumulation of new viral particles that will be released. And in that case, what you have, it's all val proteins are expressed, and ultimately the cell will die, either by the virus or because of the immune response. Okay? And so just for the purpose of the talk, I highlighted those in red because we will be talking about these proteins a little bit later. So how do we study KSHV and infections? I mean, KSHV, it's a host-specific. So essentially, you can only infect human cells. Okay? And I mentioned to you that B cells are the main cell that is infected in vivo by this virus. But for a really long period of time, actually for like more than 10 years, investigators on the KSHV field have been trying to infect B cells without any success. You could find every other cells. You can infect every other adherent cell that you want, but you could not infect B cells. And it's only 2.5 years ago, 3 years ago, that we managed to infect B cells. And essentially, one thing that really helped the field is realizing that tonsils could be infected. I mean, you can isolate B cells from tonsils, and you can infect those B cells. So now in lab, what we receive, it's tonsils from children that have been under surgery, and they send us the tonsils. You can dissociate the tonsils. You can isolate the B cells or the T cells, or can infect them as a mix. And the trick for infecting those B cells is to use co-culture. And you will co-culture those B cells with adherent cells that are lytically expressing the virus. Okay? And what you need is cell contact between those cells and the B cells to allow for the B cells to be infected. If you take cell-free virus, those cell-free virus cannot infect B cells. You need cell-to-cell adhesion, cell-to-cell interactions. And if you want to know a little bit more about that, you can see, Alice poster later on. So essentially, that's how you can infect those B cells. And you can monitor infection, in our case, by monitoring val proteins, but we can also look at GFP. And essentially, in that system, what we have B cells that are infected and are latently infected. And you can see here the kinetics of those B cells, and here it's GFP, those are gated on B cells. And you can see that here there is uninfected cells, and you can see a progression into the infections. Okay? Importantly, those B cells, those increased level in infections, are not that the cell proliferate more once they're infected. We haven't seen that. It seems that it's really continuous infection of those B cells. So those B cells, I don't have all the technology that Anne Irving has, so we could not stain many, many markers on those B cells, but essentially they are naive B cells that are infected. And it's been looked by others, by the group of Dinkidis, essentially. And it's essentially cells, it has been shown that these naive B cells, AGM B cells, essentially carrying lambda light chain and never kappa lab chain for a reason that people don't really understand. But essentially, B cells get infected. We could see also abortive T cell infection in that system. So now that we could infect B cells, we can look at how KSHV interact with those B cells and what val proteins or what cellular proteins can be affected by the infections. And essentially, when we did that, what we were really interested in is to observe that there is a protein called Activation-Induced cytidine Deaminase, or AID, and we showed that those proteins were highly upregulated in those infected B cells, and also initially on those T cells that were infected, really surprisingly. Okay? And we were interested into that protein because it's a member of the ApoBex3G family that we just heard last talk. It's a cytidine deaminase. And essentially, it's a protein that is well known to immunologists. Okay? This protein will essentially target cytidine, and we convert the cytidine into deaminate the cytidine and convert them to an uracil. And uracil will be subjected to removal of the bases by UDG, and that will be repaired by error-prone base excision repairs, for example, or by mechanism that will ensure, or try to ensure at least, that a proper repair. And this is known to immunologists because this AID is normally tightly only expressed only on germinal center B cells. So it's a stage into the B cells where those B cells will undergo what we call somatic hypermutation and class switch recombination, which are essentially two events that are meant to improve the efficiency of the B cell response and the antibody response. That will create antibodies that are higher affinity and have the proper constant region, to to ensure that with a proper effector response is associated with this. And all those two events are associated with a specific targeting of AID into the immunoglobulin locus of those proteins. Okay? And in case of somatic hypermutation, that's what you will have. You will introduce some mutations that will be potentially selected to increase affinity of the antibodies. Or you can have also class switch recombination, which is recombination of the DNA. And those will, by several excisions by AID and UDG, you will essentially cleave the DNA and recombine two different pieces of DNA to change FC regions. Importantly, AID is not only restricted as much as we would like to the immunoglobulin regions, but it's also been shown that when AID is present, it can be induce mutations in other regions of the genomes. And it's shown that many B-cell lymphomas carried out a mutation that corresponds to an expression of AID. Why are we interested in AID? Once, because it's, in the case of our infection, is not expressed in germinal centers, expressed in naive B-cells. And there is one previous examples where essentially AID has been shown to be expressed in naive or pro-B-cells, actually, on non-germinal center B-cells that were infected by another virus, and that is Abelson murine leukemia virus. And what people have shown in that case is that AID, this infection triggered the activation of AID. AID somehow is linked to the DNA damage because that creates these mutations into DNA that activate the DNA damage. And DNA damage is associated with NK cell responses. So, and that's just to refresh maybe your memory or to tell you about NK cell recognition of target cells. So essentially, what I want to talk, it's NK cells, and those are healthy cells. And as you can see, these NK cells carried out, or like express at the surface some receptors that are inhibitory receptors and some that are stimulatory receptors. Okay? And stimulatory receptors, or inhibitory receptors, will interact with MHC class 1, which is present in every nucleated cells. And that will lead to negative signals. And stimulatory receptors interact with cellular protein that are normally really poorly expressed on healthy cells. And this means that if the NK cell encounters unhealthy cells, there is essentially only a negative signal to the NK cells, and the NK cell will ignore the healthy cells. Now, in case of infection, of transformation, or stress, by mechanism that is not fully understood yet, what you can have, it's either downregulation of MHC class 1, and upregulation of this stress molecule, of this ligand for these stimulatory receptors. And that will create a scenario where you have an overwhelming positive signal into those NK cells. So the NK cells get activated and will lead to release of perforin granzymes that will induce the apoptosis of the infected cells or transformed cells, as well as some pro-inflammatory cytokines. What's interesting in the case of the Abelson story I was just mentioning is that what they observed this upregulation of these ligands, and it showed that it was associated with AID and DNA damage. Okay? And DNA damage is something that, the fact that DNA damage activates ligand and NKG2D ligand is something that has been shown here at Berkeley by David Rollet at Berkeley. And essentially, there is many stimulatory receptors, in cells. The one that is the most understood is the NKG2D receptors. And these NKG2D receptors interact with ligands and different ligands that are cellular proteins again, MICA, OMIC-B, ULBP124, or MALT1. Okay? And in the case of Abelson, it's essentially the mouse constituent of those stress protein that were upregulated in infected cells when AID was present and only when DNA damage was associated, when present. So, okay. So what we decided to do is he's now infected B cells by KSHV. Do they upregulate NKG2D ligands? And as you can see here, we infected those cells the way I've shown you before. We did an isolated by sorting the infected versus non-infected, and we look by RT-PCR for the expression of these ligands. And as you can see here, that's uninfected and infected. Some ligands are expressed to quite levels. And if you look at protein level using FACS, you can see that infected are in red, and non-infected or uninfected are in gray. And you can see you have a strong upregulation of the ligands at the cell surface. So that really meant that we had an upregulations. And something I will not show you, but you can block these upregulations by adding drugs that inhibit specifically the DNA damage response. So essentially what we believe is the upregulation of AID in those cells activate the DNA damage response, and that leads to the upregulation of these ligands. So is it, is this physiologically relevant, these ligand upregulations? So essentially to test that, what we took, it's infected cells or cells that were not infected. We put them in contact or together with an NK92 effector cells, which is an NK cell lines that is responsive, that encode NKG2D receptors. We incubate those cells together, and we looked at death of the cells. And we measured that by FACS using Pacific blue death stain. And essentially what you can see here, those are the uninfected cells. If you go target alone, you don't see any death. And if you put NK cell, you see death, but not really significant. But if the cells are infected, you see a vast majority of death that is associated with the presence of the cells. So essentially it seems like these upregulations of NKG2D lead to the death of the cells. So, but that's essentially, important, we think. And, NK cells has been shown to be present in lesions encoding, of Kaposi sarcoma lesions, and is believed to be important in elimination of KSHV-infected cells. So that might be relevant. But what we wonder is AID expressions acting also on KSHV by other means than just creating, activating NKG2D ligands? So what we decided to do is, does AID expression impact our latency on the cells? So that's a problem because Tonsilar B-cells, they express AID, and we don't have really means to make them not express AID because we don't have transgenic humans to do that. So essentially, we turned to another system where we took BCBL1, which are cells that are latently infected by the virus. They've been isolated from a patient that has one of these B-cell lymphomas. And so those viruses carry forever now the latent genomes. And what we did, we introduced AID, or we introduced vector control in those cells. We selected those cells, and we look at whether those cells were able to maintain latency and maintain the same level of episomes in those cells. And we did that by measuring LANA expression as a proxy. And you can see here that LANA transcript are exactly expressed similarly whether you have vector expressing cells or AID-expressing cells. Or if you look at bioimmunofluorescence at LANA, which are those specific dots, you remember LANA maintained the episome tightly associated with the genome DNA. So you can see that is a nucleus, and those every spot, every speckle here, it's a dot of LANA, so probably one of those episomes in those cells. And basically you can count, and there is the same number of episomes and speckles per cells, and they all maintain exactly the same way. So latency doesn't seem to be affected. So what about reactivations? So what we did, it's essentially use the same cells. And in order to reactivate the virus, as I mentioned earlier, we don't know really in vivo how to do it. But ex vivo, what we could do is treat the cells with TPA, phoboesters, and ionomycins. We can also use HDAC inhibitors, that we just heard previously, that reactivate herpesviruses. And you can measure reactivations. And you could do that by measuring RTA expression, so this lytic switch I was mentioning earlier, the first protein made upon reactivations. You can measure the genome number copies, the number of genome copies in a supernatant of those cells. And you can also take that supernatant and infect new cells, adherent cells, and measure how many cells will be infected. So if you do that, if you take cells, you transduce them with AID, you wait a week, and you treat them. And as you can see, those are vector and AID. What you see, it's at 24 hours, you see RTA expressed. And similarly, whether they're expressing cells are expressing AID or vector controlled. And if you do that at 48 hours, you can start seeing a little bit of like a loss of RTA expression, but it doesn't appear really drastic. And you can keep those cells even 10 weeks in culture. And what you can see at that point, you can see really starting a defect in terms of reactivations. So is that meaningful? So what you could do is look at the other mean of measuring lytic reactivations. And what you can see, you can look here at the total genome copies in a supernatant of the cells. And what you can see is like, despite you have only two-fold downregulation of RTA, or threefold, what you can see is that leads to 2 logs inhibition of VAL productions. Okay? And you take those viruses, you can even infect cells with a dilution of the VAL supernatant and count the number of cells that were infected by using this VAL supernatant. And that you can see here, you have even 3 logs inhibition in terms of reactivations. So clearly, AID has an impact reactivations of the KSHV virus, and has a big impact on the outcome of the infections. We isolated these viruses. We didn't do genome sequencing, but what we sequenced some part of these viruses, and we never found any mutations, or at least an increase in mutation in the AID-expressing virus producing AID-expressing cells. So we think it's something different going on there. We don't really know yet. So as you know, AID seems to increase NKG2D ligand upregulations, and AID seems to be detrimental for the VAL reactivation as well. And as you know, viruses that have evolved with us for a really long time have had time to develop strategies to usually counteract the innate responses of the host, or responses that are deleterious for the virus. So we decided to look into that. And the first thing that we thought about was looking at uracil D-glycosylase. And essentially, that's a protein that when there is a cytidine that is being deaminated using by AID or other means that create a uracil, this protein this base is extracted by UDG, and that is being repaired. Okay, and that's a mechanism. So UDG is involved into the repair of the deamination events. Okay, and it seems like in the genomes, every cells, every day we have spontaneous deaminations, and those are spontaneously repaired by UDG, and that's maybe 1,000 of them every day in every cell that we have. What we are interested in UDG is because UDG is known to interact with LANA. And actually, LANA interacts strongly with UDG. And LANA recruits UDG to the latent genomes. And that's been observed by the group of Earl Robinson. And essentially, the reason wasn't really clear at that point, why LANA would interact with UDG and what recruit to the latent genomes. But it was believed initially that that uracil were spontaneously misincorporated upon VAL replication processes, and those where this LANA interacting with UDG was required to repair these misincorporations. Okay? So we decided to look whether UDG was involved in our case. And that just to illustrate what I was mentioning, LANA, colocalizing and interacting with UDG. You can see LANA, which has those red speckle in that case, UDG, which has the blue, green speckles, and you can see there is a strong colocalization between the two. And that works with, co-IP and all the other experiments you can imagine. So does he has an effect? So what we did is just like shRNA and UNG, or UDG, it's the same. And what you can see, even like after a week of post-transcription, when normally we have barely any downregulations of RTA and was expressed similarly in AID or vector expressing cells. In that case, if you have no UDG, or we have about 50% downregulations, you can see that you have a drastic effect on the ability of the cells to reactivate. So clearly, UDG is involved in our case, and probably through LANA, but that's difficult to prove in that case. So is it the only mechanism? And how AID is usually regulated? I mean, you can regulate the effect of AID using UDG. But what is known also is that microRNA tends to regulate AID. And there is human microRNA that are known to specifically interact with AID mRNA and regulate AID expressions. And one of them is miR-155. So that's a human microRNA. It's expressed, same thing, conserved into many species and to lamprey. It's conserved, so it's not only involved into those, regulation of AID. This microRNA is actually a key regulator in immune function, so it's not only regulated AID, it's also regulating, for example, repressing SMAD5, which result in RB inactivations and proliferation of the cells. So it's inducing proliferation, and that's why miR-155 would be expressed in many lymphomas, because it promotes proliferations, but other cells as well. What has been clearly, definitely shown as well, it's like the lack of miR-155 it's been linked to an increase in AID expressions and can increase translocations, which are normally associated with AID. So that's the alignment between the human AID, the 3' UTR of AID, and miR-155. And if I'm talking about these microRNAs, it's because within the KSHV microRNAs, the 17 that are encoded by KSHV, there is one called K12-11 that is also an homolog of miR-155. And actually, this you can see here, there's really a seed sequence bind with the same, it's exactly the same seed sequence with miR-155. And actually, experiments in mice have been shown that you can remove miR-155 and replace it by K12-11, and you will completely restore every defect that you observe in that case. So we look if that could regulate AID, and if any other of those microRNAs could, in fact, regulate AID. So we did a bioinformatic screen. We look at, by bioinformatic, which of these KSHV microRNAs can interact with the 3' UTR of AID. Those KSHV microRNAs are involved already in some other function that I don't want to talk about now. But essentially, we found non-microRNAs that potentially could interact with AID. We cloned to test whether these microRNA were indeed able to regulate AID expression. What we did is clone the 3' UTR of AID downstream of the luciferase genes. So it's you can transfect that into cells together with microRNAs, and you can measure the efficiency of those microRNAs to prevent luciferase expressions. And what you can see is that K12-11 as well as K12-5, which are those two here, as well as miR-155, the human one that downregulate AID, those three downregulate the expression of the luciferase. And those are the location of those microRNAs where they're supposed to bind on the 3' UTR of the AID mRNA. So just to go fast now, you can see that together they downregulate slightly better. That's miR-155, the human one, K12-5, K12-11, and that's the sum of the two. If you can transfect both of them, and they're downregulate slightly better. You can mutate, you can define the sites where K12-5 and K12-11 bind on AID, and you can show that as predicted, that's the one that I showed you before. So that's the one that were predicted. So to test if those were microRNA mimic, so they express to really high levels, to see if the endogenous level were efficiently able to inhibit AID, what we did, we cloned shRNA coding for those microRNAs in cells, and we compared the level of expression of those microRNAs with cell lines that have been isolated from patients carrying the virus latently. You can see BCBL1, BC1 here, and you can see that K12-5 you express in the cells. We have exactly the same expression. K12-11 expressed at the same level as well, and that's luciferase, so of course it's not present on the luciferase cells. And you can now look on the AID. If you transfect AID on the cells, you can see that AID is reduced, expression of AID is reduced in cell expressing K12-5, K12-11 that are expressed at physiological level. You don't see much mRNA degradation, so it's probably acting at the translation level. So is it acting within the context of the viral infection as well? So what we did is to KSHV microRNAs. We took cells expressing either the full-length mRNA for AID or just an mRNA that were expressing the AID, no 3' UTR in that case. We infected those cells with viruses that express wild-type KSHV or virus that lack all the microRNA regions. And what you can see, I go really fast, you can see that here it's that the one that, in red, it's a virus that expresses microRNAs, and in gray, it's a virus that don't express microRNAs. And you can see that those inhibits only the full-length mRNA, but you don't see any difference when you don't have the 3' UTR of AID. So what I've shown you, it's AID is upregulated in infected cells. We don't yet know by what molecular mechanisms. Is it DNA sensing? Is it a viral protein that it expressed early on during the viral cycle that is responsible for AID expression? We think the later it's the case. We transfected VRF2, which is a non-protein KSHV protein activating NF-kappa B, and that leads to the activation of AID in cells. I've shown you that AID expression negatively impacts KSHV reactivations. I've shown you as well that UDG is required for reactivations in the presence of AID, is really controlled. And I've shown you that the virus encodes miRNAs that decrease the AID expression in infected cells, probably to allow for some degree of reactivations. So this has been done by Elena Beckerman, which is somewhere in the audience. So I told you a questionnaire for her, and I don't think I have time for any. Those are the people that have been involved. Michele has been a member of the Role lab, and he's been helping Elena to do the NK killing assays. And the collaborators were Adam, which done the bioinformatics for us, SJ Gao, who give us the viruses lacking microRNAs. And David has been really helping us for the last years, many years. Is the one working on NK cells, and discover NKJ2D and the expression of those ligands and DNA damage inducing those ligands. So he's been a key member of this group. So thank you very much, and I'm open for questions. Yep. Okay, let's go to, yeah.
Oh, sorry. Go ahead.
Okay, yeah.
I was wondering if you somehow modulate the NKJ2D levels to affect clearance.
We don't know. We're looking into that by other mechanism. We're looking at the HCMV and MCMV infection, and we show that HDAC actually inhibitors, and it's been shown by David Role and others as well, are stronger regulators of those NKJ2D ligands. So it might be one way that people want to explore later.
My question is, if you just transfect DNA, do you see upregulation of AID, like into cytoplasm or into?
We see it in Tonsilar B-cells, so it's really hard to do in Tonsilar B-cells because those are primary B-cells, really hard to transfect, really hard to work with. If you transfect vector, using AMAXA technology, we've been able to transfect DNA. If you transfect vector, you don't see AID upregulation. If you transfect VRF2, which is one of the validly encoded proteins, that is really expressed early on upon the life cycle, you see upregulation of the NKJ2D ligands. And that one trigger NF-kappa B, so that's what we think NF-kappa B is involved in those cases.